You know those incredibly complex life-saving medicines, things like antibody therapies? Ever wonder how they're actually made? Well, the answer isn't in some chemistry lab. It's inside a microscopic biological factory, the cell. Today, we're going to go on a little scientific quest to see how we've learned to command these tiny factories to produce the proteins that are literally shaping modern medicine.
And really, it all boils down to this one big question. To create the medicines of tomorrow, we have to solve this massive biological engineering puzzle. How do we get a cell to produce one specific protein, not just correctly, but in absolutely enormous quantities?
All right, let's dive in. Here's our game plan. We'll start by looking at the first big hurdle scientists hit. Then, we'll see how they found a much better solution and came up with some seriously clever tricks to boost production. We'll also cover how they pick the right tools for the job, and finally, we'll get a little peek into the future of this amazing technology. So let's rewind. In the early days, the plan seemed pretty simple, right? Scientists needed a factory that could grow super fast and take instructions easily. And their first choice was the classic workhorse of microbiology, simple bacteria like E. coli. I mean, on the surface, bacteria looked perfect. They're simple, they replicate at lightning speed, and they're pretty easy to work with. You compare that to the much more complex, slower-growing mammalian cells, and using bacteria as your protein factories, well, it looked like a total no-brainer.
But here's the catch, and it was a huge one. While bacteria were great at reading the genetic blueprint and, assembling the basic protein chain, they were like a very basic assembly line. They just didn't have the sophisticated, high-tech tools needed for the crucial finishing touches on complex human proteins.
So what are these finishing touches? Well, in biology, they're called post-translational modifications. Think of things like glycosylation, where the cell adds these specific sugar molecules, or phosphorylation. Now, these aren't just little decorations. They are absolutely critical instructions that tell the protein exactly how to fold into its precise 3D shape. Without them, you don't have a functional protein. You just have a long, useless string of amino acids. So, with bacteria out of the running for these complex jobs, scientists had to find a new hero. And that's when they turned to the mammalian cell. It might be slower, yeah, but it was the sophisticated, high-tech factory they needed all along. And the reason they work so well is actually pretty simple. They're basically our own cells. They already have the entire suite of advanced machinery built right in. They instinctively know how to do all those complex folding and modification steps, making sure the final protein, like say a complex antibody for cancer therapy, is fully functional and works exactly like it's supposed to in the human body. Okay, so we've got the right factory, awesome. But now the challenge is scale. How do we get that factory to produce our protein not just correctly, but in huge, huge amounts? Well, the first strategy is to make the genetic instructions a lot louder and clearer. The key to this is a little piece of DNA called a promoter. And honestly, the best way to think of it is as a volume dial for a gene. A weak promoter, the gene is barely whispered. You get very little protein. But a strong promoter, that cranks the volume all the way to max, telling the cell's machinery, read this gene, read it over and over and over again. And to really crank that dial, scientists got clever. They decided to borrow super powerful promoters from viruses, like SV40. Think about it. Viruses are masters at hijacking cell machinery to make tons of copies of themselves, so their promoters are incredibly strong. By sticking a viral promoter onto our gene, we can totally supercharge protein production. So, turning up the volume on one blueprint is great. But what if we could fill the factory with hundreds of copies of the blueprint itself? Now that is the next brilliant strategy. This is done through this amazing process called gene co-amplification. The core idea is just brilliant. You trick the cell into making tons of copies of your gene by linking its fate to another gene that's absolutely essential for the cell's survival, but only under very specific stressful conditions. Okay, the classic example of this is the DHFR system, which is used in what are called Chinese hamster ovary, or CHO, cells. These are the absolute powerhouses behind blockbuster drugs like Humira. Now, here's the clever part. Scientists use a vector to insert two genes into the cells, their target gene and a survival gene called DHFR. Then they add a drug that would normally kill the cells by blocking DHFR. The only way for a cell to survive this onslaught is to make way more of the DHFR protein. And the only way to do that is to make hundreds of copies of the DHFR gene. And because our target gene is physically attached to it, it gets copied right along with it. It's genius. And the power of this system is just incredible. We're not talking about 2 or 3 extra copies here. Scientists can select for cells that contain up to 1,000 copies of the gene. This is literally how they create these hyperproductive supercells that become our protein-making powerhouses. But, maximizing production isn't just about clever genetic tricks. The DNA blueprint is critical for sure, but the factory you put it in, the cell line itself, is just as important. As one of the sources for this puts it so well, the DNA vector is only half the equation. The cell line provides the other half. You can have the best blueprints in the world, but if your factory floor isn't equipped for the job, your output is going to suffer. It's a true partnership. And just like in manufacturing, right? Not all factories are the same. Different cell lines are specialized for different tasks. So cost cells, for example, they're engineered to make copies of the DNA blueprint really, really quickly. So they're great for rapid, early-stage tests. But when you need to scale up for reliable, high-yield production, you turn to the industrial workhorses like C127 or those famous CHO cells, which are optimized for stability and just massive output. So the final choice really comes down to a lot of practical questions. What kind of protein are you making? How much of it do you actually need? What are the costs going to be? And just as important, what are the safety and regulatory hurdles you have to clear? It's this complex calculation for every single product. The techniques we've talked about are incredibly powerful, but you could almost say they're a bit of a blunt instrument. So now, let's look at the cutting-edge tools that are bringing a whole new level of surgical precision to this process. The future is all about precision engineering, and its superstar is, without a doubt, CRISPR. If the old methods were about adding new pages to the cell's instruction manual, CRISPR is like having a find and replace function for DNA that lets you edit a single word on any page you want. It's a total game changer. So with CRISPR, scientists can go in and precisely turn off a gene that might be slowing down production, or they can introduce very specific, targeted edits to the cell's own DNA. We're moving beyond just adding foreign blueprints. We're now able to re-engineer the factory itself for maximum efficiency. So think about it. We've gone from struggling to make a single functional protein to being able to precisely rewrite a cell's genetic code to turn it into a bespoke biological factory. And that just leaves us with this final, tantalizing thought. Now that we can build almost anything, what will we choose to build next?